Okay, here's Chapter 49, Sensory and Motor Mechanisms. As an introduction, we have action potentials, and you need to know the difference between sensations and perceptions, which are written down on the slides. Alright, sensory reception, it's written down, and you need to know the difference between exteroreceptors and interoreceptors. Intero being inside the body, extero being outside the body.
There are 4 types of functions for receptor cells: sensory transduction, amplification, transmission, and integration. Know the difference between those.
Alright, there are different types of sensory receptors: mechanoreceptors, pain receptors, thermoreceptors, chemoreceptors, and electromagnetic receptors. Mechanoreceptors being like touch, pressure, that
sound.
kind of stuff. Pain receptors being pain, thermoreceptors being hot and cold, chemoreceptors being like taste and smell, and electromagnetic receptors being like photoreceptors seeing light, feeling electricity. There's one more. Magnetism.
Photoreception. In invertebrates, there are these eye cups, so in flatworms, say, for instance, and they're the simplest type of eye. They have only light intensity and direction, and they evolved to form compound eyes, which are found in insects, crustaceans, flies. They consist of several thousand light detectors called ommatidia, so, for example, just a fly, and a single lens eye, which is found in jellies, spiders, and mollusks, and they're camera-like, so the retina moves and the lens moves like a camera.
Okay, and here's the structure of our eye for a vertebrate. It's very similar to the single lens eye. Diagram's a good thing to know. Alright, you need to know the difference between rods and cones. Rods contain rhodopsin, and they see in black and white. They register light. Cones contain photopsin, which is a visual pigment, just like rhodopsin, and they see color. And there's 3 different kinds of cones: red cones, green cones, and blue cones, depending on the type of photopsin. And there's the fovea, which is the center of vision, which has a dense cone accumulation. You see the best there.
So you see light in cones, and black and white you see in the dark rods.
Okay, so here's rhodopsin. Rhodopsin is a visual pigment in rods, and it's made of retinal and opsin. Retinal being the pigment and opsin being the membrane protein. Together they make rhodopsin. Okay, so what happens here is when light hits rhodopsin, it's activated, and an activated rhodopsin brings in transducin, which activates phosphodiesterase, which is an enzyme. Okay, and then here you can see there's sodium channels, and what's holding open these sodium channels is cGMP. The phosphodiesterase that was activated turns cGMP into GMP. Doing so, the sodium channel is closed. Upon closing the sodium channel, you have hyperpolarized this cell, and hyperpolarizing that changes the membrane potential, and by changing the membrane potential, you create an action potential, which is sent through the nerves,
thus enabling you to see. In processing visual information, there are these things called bipolar cells, and they're basically synapses made of rods and cones with the neurons, and ganglion cells, which transmit action potentials to the brain via the axons, and the horizontal and amacrine cells. So on the diagram on the next slide, light enters your eye and goes to the retina, and in the retina, light hits the ganglion cells, which make a signal to the amacrine and horizontal cells, to the photoreceptors, to the brain. So basically it's just another pathway just to the brain, how it does that. Hearing and equilibrium. In hearing, there are 3 parts of the ear: the outer ear, the middle ear, and the inner ear. In the outer ear, there is this tympanic membrane, which is also called your eardrum, that vibrates with the same frequency as sound. And in the middle ear, there's these 3 ossicles, or small bones, called the malleus, the incus, and the stapes. And so when the tympanic membrane moves, it moves the 3 ossicles, which magnifies the sound. And so that goes into the inner ear, stimulates the cochlea, which converts movement into signals to the brain. The oval window is basically just like when it's pushed, it creates a wave to the cochlea, and the eustachian tube just equalizes the pressure between your ear and the atmosphere. So, for example, when your ears pop. The organ of Corti just contains the receptor cells of the ear, or the hair cells, and the round window just allows movement of fluid in the cochlea for equilibrium. Equilibrium. Behind the oval window is a vestibule that contains two chambers, the utricle and the saccule, which respond to changes in head position with respect to gravity and movement in one direction. So there's little hairs in the utricle and the saccule that gravity is always pulling downward on it, so you can always tell where you are for gravity. And the semicircular canal contains fluids, basically, that so whenever you move, it can tell whether or not the angle changed or not. So when you're dizzy, you move your head, and then the fluid continues to move while your head stops. So that's what makes you dizzy, basically.
Yeah, the next slide contains a diagram that shows you these three parts.
In the lateral line system in aquatic amphibians and fish, the water moves through the body of the fish and hit the neuromasts, which are basically the cupula, and they also contain the hair cells. So when the water moves past them, the hair cells generate an action potential to the brain.
Okay, so invertebrates have statocysts to maintain equilibrium, and they contain mechanoreceptors. And what they have is they're like circular things, and they have cilia on the inside and statoliths. And statoliths are like grains of sand. So what happens is when the animal moves, the sand moves in against the cilia, and when the cilia is stimulated, it goes to the ciliated receptor cells on the outside, and that senses their balance with the sensory axons. Also, insects have body hairs on the outside of them that vibrate in response to sound waves, which is kind of like how they would hear.
Chemoreception. The perceptions of taste and smell are usually interrelated. So you have your pharynx, which has three parts. There's the nasal pharynx and the oral pharynx, which is in your mouth. The nasal pharynx is in your nose, and your regular pharynx.
Okay, so taste buds are the receptors for taste, and they're modified epithelial cells on the tongue and in your mouth. So this diagram shows a sugar molecule, so you ate like a cookie or something, and sugar molecule attaches to your sugar receptors, the taste buds that register sweet things, and the diagram zooms in on a neuron here, a sensory receptor cell, and what happens is when the sugar molecule attaches to the receptor, it activates this G protein, which activates phospholipase C, which takes this IP3 from the PIP2, separates it from the phospholipid bilayer, which then activates this calcium ion channel, which then goes over here and then opens up the sodium channel and lets sodium in, which then changes the membrane potential of the cell, which then sends a action potential through the sensory neurons, and you taste stuff.
Basically another signal transduction pathway. Smells. So when you smell something, there are odorants that go into your nose. Then there are odorant receptors which accept those odorants. And the odorants also go into your nasal cavity. And there are cilia in your nose that make a signal to the chemoreceptors, which then go to the bone, to your olfactory bulb. And that's basically where it sends action potentials to the brain, allowing you to smell. Movement and locomotion. In locomotion, basically is just, movement is basically just the contraction of muscles working against a type of skeleton. So, in swimming, overcoming gravity is no problem because, it's underwater. But the problem is water is much denser than air, so there's this problem of resistance. So that's why you see a lot of animals or fish have fins that can help them move around. In land, air poses little resistance, so there's nothing really there to move against it. But animals must be able to support itself and move against the gravity. So kangaroos have really big feet that allow them to jump long distances. And in flying, gravity is a problem, so you have your light birds with their light bones and their feathers that propel them through the air really quickly. In a skeletal and cellular level, there are microtubules, or cilia and flagella, that can move, and microfilaments, which allow muscle contraction and movement in amoebas.
Okay, so there are three types of skeletons. There's a hydrostatic skeleton, which is fluid held under pressure in a closed body compartment, and it moves through peristalsis. So it's like a worm that like contracts in one area, so it gets like uh, thinner, and then but it gets wider in the back, and then that's how it moves along like that. And then there's an exoskeleton, which is a hard encasement deposited on the surface of an animal, so like a beetle with a really hard shell. And then there's the endoskeleton, which was what we have. Hard supporting elements such as bones buried within soft tissues of an animal.
So there are two types of muscle filaments, thick and thin. Thick filaments are made of basically myosin, and thin filaments are made of three parts, troponin, tropomyosin, and actin. We'll get into that very soon.
Okay, so the sarcomere is a basic contractile unit of muscle, and it's separated into different parts. There are the Z lines, which are the borders of the sarcomere, and they're just thin filaments. And the I band is the part of the sarcomere that is only thin filaments, just actin. And then the A band is the broad region corresponding to the length of the thin filaments. So it's not like the thick filaments themselves, it's just the width of, or the length of the thick filaments. And the H zone is the center of the A zone that contains only thick filaments. Okay, so the T tubules are what allow action potentials to spread deep into the interior of the muscle fiber. So when your brain sends an action potential, like we want to contract this muscle, then the T tubules are what send those into the muscle. And then the sarcoplasmic reticulum is what regulates and releases calcium ions, which becomes important in the next slide.
So in thin filaments, there are three components, the actin, tropomyosin, and troponin. And tropomyosin basically just binds everything together in the actin. Troponin basically blocks the myosin binding sites. So thick and thin filaments bind together from cross bridges, but troponin inhibits that. So it binds, when calcium enters the muscle through the calcium channels, it binds to troponin, which makes it basically kind of just go away. So then it allows the myosin to bind to the actin, and that's when muscles contract. So they overlap together, and they just contract the muscle. So you have your thick filaments, and in these thick filaments there are these cross bridges. That's where it binds to the thin filaments. Your myosin head is on your thick filament, and it's like a little bit of a pedal that can move or scooch the thin filament together, and basically that's what makes it overlap. And there's a little bit of a video down below if you need more clarification.
Okay, so tetanus, which, people get tetanus shots. Tetanus is caused by a bacteria, which you get the shot to get rid of it. And what happens is you can no longer prevent those action potentials from showing up at your muscles. You don't ever relax your muscles, which is like when you get lockjaw and it's stuck that way, your muscles are completely stuck, contracting. And yeah, that's what tetanus is. You don't want it.
More body movements. So a motor unit is a single motor neuron and other muscles that it controls. So basically, a motor neuron has all these muscles, and that unit is just that group. And so when a recruitment is basically just when the number of motor neurons increase. So when you have tension in your muscle, it's increased because it's activating more motor neurons that control that muscle. And there are two types of muscle fibers: the fast muscle fibers, which are the rapid, powerful contractions. Example: flight muscles in birds, because they have to migrate so fast and long, which is oxidative, so it relies on aerobic respiration to generate the ATP, or glycolytic, which uses glycogen as a source of ATP. And then there's also slow muscle fibers, which are sustaining long contractions, maintaining the posture. So for instance, if I'm standing up, it's using a slow muscle fiber, and that's oxidative, so it uses breathing or aerobic respiration to generate the ATP.
Okay, so in conclusion, you guys need to be able to examine input and output of the coordinating system that controls so many aspects of animal behavior. You need to know how sensory receptors work: taste, smell, sight, hearing. You need to be able to examine the structure and function of muscles and how motor units work and sarcomeres and fun stuff like that. And you need to know the different skeletons and how they work in the context of body movement.
Good luck and have a nice day.
That's chapter 49.